Okay, let's go.
One of the coolest things is that each of their 8 arms has its own mini brain, plus a central brain. It's like if your limbs could think for themselves while your main brain just supervised.
What are we talking about?
Octopuses and their 9 brain setup, how their arms basically have a mind of their own. Want to keep going with that? Or pivot?
Okay.
So get this. When an octopus is exploring a crevice or grabbing food, the arm doesn't wait for instructions. It acts independently, sensing and reacting on its own. The central brain might not even know exactly what the arm is doing in real time. Isn't that wild?
It's kind of wild. All right, let's formulate a set of questions we can ask about animals, kind of like that one. I want you to ask me those questions.
Totally. Let's build a vibe of questions that spark curiosity. Here are a few to kick us off. Why do some jellyfish seem to live forever? What's the deal with electric eels? How do they shock things without frying themselves? Can pigeons actually do math? How do bats see with sound? And is it better than eyesight? Why do flamingos stand on one leg? And does it actually save energy? Want to keep that style? Or lean more into the weirder or darker side of animal behavior?
Okay, now ask me questions about why do flamingos stand on one leg?
All right, let's dive in. Why would an animal choose to balance on just one leg for hours? Doesn't that seem harder? Not easier?
Yes, it does seem harder. It seems like the balance would be an issue. But I guess somehow they must solve the balance issue somehow and make it low energy to stabilize. And then why do they actually only use one leg? Is it somehow more efficient to only use one leg instead of two? Seems like at least naively, you would think you're going to use more energy stabilizing on one leg than you are stabilizing on two legs. So, yeah, I don't know the reason that they only use one leg.
Exactly. And that's what makes it so fascinating. The weird part is, studies have shown that flamingos are actually more stable on one leg than two. When they're on one leg, their body kind of locks into position. It's a passive stability, almost like a standing gear. No muscle strain. So then the follow up is, why evolve to do that? What's the advantage of standing like a yoga master in a bird spa?
Before we do that, explain the stability. It must be a biomechanical thing. You have access to papers on the internet and things. Why is standing on one leg actually more stable than standing on two?
Flamingos have a unique locking mechanism in their legs. When they stand on one leg, their joints naturally align, allowing gravity to hold them in place without muscle effort. This passive stability makes standing on one leg less tiring than standing on two.
Okay, so you're saying they just lock everything and stand on one leg? They don't have to be actively stabilized?
Exactly. It's almost like their leg clicks into place. When a flamingo stands on one leg, its body weight shifts in such a way that the joints line up vertically, forming a stable column. Researchers even found that dead flamingos can stay balanced this way. No muscles engaged at all. It's pure passive mechanics.
Okay, maybe that has something to do with it.
Yeah, and that passive balance might be just one piece of the puzzle. The next question is, what's the payoff? Like, even if it's easy to stand that way, why bother using only one leg at all? What's the benefit?
Yeah, why not just lock two legs? I bet most birds do, actually.
Right? If you can lock one, why not lock both and be a pink tripod? One idea is thermoregulation. By tucking one leg up, flamingos reduce heat loss through their limbs, especially since they often wade in cold water. Blood vessels in bird legs are close to the skin, so they lose heat fast. Keeping one leg out of the water cuts that in half.
Okay. So, comparatively, what do other birds do when they're just standing for long periods?
Great question. A lot of birds actually do the same thing. Herons, storks, even ducks will sometimes stand on one leg. It's especially common in species that spend time in water or cold environments. But what's different with flamingos is just how often they do it and how stable they are. Most other birds still rely on muscle control for balance. Flamingos take it to the next level with that mechanical lock-in, like a built-in kickstand.
I mean, like birds that live in trees.
I got it. Perching birds, like songbirds or parrots, they've got a totally different setup. Those birds have a perching reflex. When they land on a branch and bend their legs, tendons in their legs and feet automatically tighten and curl their toes around the branch. It's like a ratchet. They physically can't let go until they stand up straight again, so they can sleep perched without falling off. It's kind of the tree dwelling version of what flamingos do in water. Both use passive mechanics, just tuned to their environment.
All right. Next question.
Why do some frogs let themselves freeze solid in the winter and somehow come back to life in the spring?
Well, probably because they just don't want to deal with the fact that there's not much food
Right?
in the winter. I mean,
Frogs, for example,
that's
survive
why.
being frozen. Heart stopped, no breathing, totally solid. The crazy part is, they don't just tolerate it, they're built for it. So what's going on inside their bodies that lets them do that without dying from ice damage?
First, explain why things die when they get cold.
Perfect starting point. Most animals die from freezing because ice crystals form inside their cells. And that's a huge problem. Ice expands and shreds the delicate membranes. Plus, freezing pulls water out of the cells, dehydrating and damaging them. On top of that, blood stops flowing, oxygen drops, and cells basically suffocate or go toxic. So normally, freezing equals chaos at the microscopic level. But not for these frogs.
What about at the molecular level inside of the cell? I mean, regardless of the membrane, which you said could be a problem if it blew up. What about all the molecular machinery inside of the cell? Could that get messed up?
Yeah, that's a huge concern too. Inside the cell, you've got proteins, enzymes, RNA, all these delicate structures that depend on precise shapes to work. Freezing slows everything way down, but it also risks denaturing proteins, making them unfold or clump up. That's bad. Plus, as water freezes outside the cell, the inside gets super concentrated. Solutes like salts and metabolites build up, and that chemical imbalance can mess with pH, charge, and structure. It's like turning the cell into a briny slush full of half-functioning molecules. Definitely not ideal for survival.
Can you freeze a human embryo safely?
Yes, you actually can, and it's done all the time in fertility clinics. Human embryos can be frozen and later thawed for implantation with good success rates. The key is how they're frozen. It's not just tossing them in a freezer. They use a process called vitrification, which turns the liquid inside the cells into a glass-like solid without forming ice crystals. That avoids all the cell shredding damage we talked about. Cryoprotectants are used, special chemicals that help protect cells during freezing. So yeah, embryos handle it surprisingly well, as long as the freezing is super fast and controlled.
What's the cryoprotectant used for embryos?
For embryos, the most common cryoprotectants are dimethyl sulfoxide, DMSO, and ethylene glycol, often used in combination with sucrose. Here's what they do. DMSO and ethylene glycol penetrate the cells and replace some of the water, lowering the chance of ice crystal formation. Sucrose, which stays outside the cells, helps draw water out gently to dehydrate them a bit before freezing, which also reduces ice risk. The balance is tricky. You need enough cryoprotectant to protect the cells, but not so much that it becomes toxic. That's why vitrification happens fast, so cells don't sit too long in these chemicals.
What kind of experiments did scientists have to do to identify these chemicals that don't hurt the cell?
They had to do a lot of painstaking trial and error experiments, starting back in the mid 20th century with both animal and human cells. Researchers would expose cells to different chemicals, freeze them, then thaw and see what survived. They tested things like how well the cells maintained membrane integrity, whether they could divide afterward, and whether organelles looked normal under a microscope. They also measured ice formation using microscopy and calorimetry. It took a while to figure out that some chemicals, like glycerol, DMSO, and ethylene glycol, could sneak into cells and prevent ice crystals from forming, either by depressing the freezing point or by helping water form a glass instead of sharp crystals. It wasn't just about survival either. They had to show the cells still functioned afterward. That meant viable embryos developing, sperm fertilizing eggs, and stem cells differentiating. So it was a long path of biochemical sleuthing, one tweak at a time.
Were there any chemicals that caused defects in the organism?
Yes, definitely. Early on, some cryoprotectants were either too toxic or subtly damaging. For example, glycerol, which was used in early sperm cryopreservation, worked well for some species, but caused problems in others, like membrane disruption or reduced fertility post-thaw. Even now, DMSO is effective but can be toxic at high concentrations or with prolonged exposure. It can interfere with cell signaling, gene expression, and even damage DNA if not carefully managed. Some studies found that embryos exposed too long to DMSO or ethylene glycol had lower implantation rates or developmental abnormalities, especially if the exposure was during sensitive stages. That's why modern vitrification protocols are like choreographed dances, precise timings, temperatures, and chemical combos. The wrong step can mean subtle defects that don't show up until later development. So yeah, researchers had to walk a tightrope between protection and poison.
What are the odds of the freezing process introducing a birth defect?
The odds are very low, but not zero. Large-scale studies of babies born from frozen embryos show that the risk of birth defects is about the same, or sometimes even slightly lower than with fresh embryos. Most estimates put the overall risk at around 2 to 3%, which is right in line with the general population. Some early concerns about things like preterm birth or low birth weight have mostly faded with improved techniques. That said, there are a few nuances. Some studies suggest a slightly higher chance of certain epigenetic changes, like differences in imprinting, which can affect gene regulation. But even those are rare, and so far there's no strong evidence that vitrification itself introduces major risks when done properly. So overall, pretty reassuring, but scientists still monitor outcomes carefully, especially as technologies evolve.
Can the process scale to larger organisms? What's the limit at which it doesn't scale?
That's the big challenge. Scaling cryopreservation to larger organisms runs into some nasty physical limits. The main issue is heat and water distribution. In small cells or embryos, you can cool and warm everything quickly and evenly. But in bigger tissues or whole organs, you get uneven temperatures. Some areas might cool slowly, forming ice. Others might thaw too fast, causing thermal stress or cracking. That's especially true during rewarming. which is actually harder than freezing. Then there's cryoprotectant penetration. In big tissues, it's hard to get the chemicals evenly distributed. You might protect the outer layers, but the core could still freeze and get damaged. Plus, high cryoprotectant concentrations can be toxic if they sit in one place too long. In terms of scale, organs like kidneys or hearts are the current frontier. Scientists have managed to vitrify and rewarm some small animal organs, but doing it reliably and without damage remains super tricky. Whole body freezing, that's sci-fi for now. There's no known way to cool and rewarm a human body evenly, prevent ice damage, and avoid cryoprotectant toxicity all at once. So somewhere between an embryo and a solid organ lies the edge of what's currently possible.
Explain exactly what you mean by glass rather than crystal.
Great question. This goes right to the heart of vitrification. When we say glass instead of crystal, we're talking about the structure of how molecules are arranged when frozen. In a crystal like ice, water molecules snap into a rigid, repeating lattice. That's orderly, but it's sharp. And those growing edges can puncture cell membranes or disrupt proteins. A glass, on the other hand, is disordered. In vitrification, water cools so fast that the molecules don't have time to line up into crystals. Instead, they freeze in place randomly, like a liquid that got stuck in motion. That's why we call it amorphous. No repeating pattern. So glass is solid but not sharp. It avoids the slicing and shifting damage that crystals cause. That's the magic of vitrification. It freezes biology without freezing it in the traditional jagged way.
Would you say it's the opposite of annealing?
Yeah, that's a really interesting way to put it. And in a sense, yes. Annealing is all about slowly cooling something, giving molecules time to settle into an ordered, low energy state. In metals or crystals, that means reducing defects and forming a nice, clean lattice. It's controlled, structural, and relaxed. Vitrification, by contrast, is rushed. You cool so fast that the molecules freeze in whatever messy position they're in, locked in place before they can organize. So instead of order, you get frozen chaos, but stable chaos. So yeah, you could say vitrification is the anti-annealing. One's all about order, the other's about preserving disorder.
Is this why flash frozen foods are supposedly better?
Exactly. That's the same principle at play. When food is flash frozen, it's cooled super quickly, so fast that ice crystals